morning, and I think today is a very interesting session as presented by the earlier scientist, my colleague Anurag. He is also working and on some aspects of the biomarkers for the early detections. And my group have yet started that how the circulating tumor cells can help for early detections by using biomolecules, by DNA and EMT markers, and no doubt by signaturing of the mRNAs. So I'm sharing with my group of the work. So the main thing, how it is progress and who is responsible. We are living in electronics, gadgets, medias, exposure with radiations, but still we did not reach into the conclusion that what are the exact factors which are responsible for transformation from normal to the malignant cells. So if you will see the figures, then what people think that the cancer is there, it is very difficult to survive. No, it is challenge to everyone's life. And they are still surviving. So we have to learn more from, if you will look, and of these figures of different professions. So no doubt, the genetics is a main component of the transformed cells. But the epigenetic factors is also to be identified that how they modified the signaling pathway, including my friend says that the P53. P53 is also known as the master of the genes. I am sharing my work especially for the younger scientists to be motivated and come forwards. So these are the mechanisms that how the genetics and epigenetic factors causes the mutations, either the double-handed or the single-handed, and modifies the proliferative markers. Because there is some difficulty arises when we start a study of the cancer cells. In this, the cancer cells is heterogeneous in natures. So you can see the stem cell. It is not working? Yes. So it is transformed either self-regulatory or by the combination of the paracrine fashions. So these are the complicated pathways, and still sometimes I myself is confused that how many molecules are interconnected, whether the stem cells are there, P50 they are there, or certain growth factors present inside the cytoplasm or the membrane or inside the nucleus. So it is a very complicated mechanism. When I started to prepare my presentation with my students, then they say that in the one-sided, the stem cell marker like OCT, NANONG, and SOX2. And they goes to the interconnected through P53. This is a master genes which regulate all the phenomena of the single transition mechanisms. But how they make a web? They make a web inside the cells. And it is very difficult to understand, but it is very difficult to, if you perform in the laboratory exercise. So, but scientists always tries to improve the management for the survival of the patients. By early genetic screening is most important aspects if you feel we or the clinicians suffer or help for the correct diagnosis. The correct diagnosis is a very important aspect. And then you have to decide whether the disease is either familial or sporadic in nature. If it is familial, then the management will be entirely different. And then accordingly, with your guidance of the clinicians, you can reach into the conclusions what type of management is required. So this is the what causes delays in the cancer of the diagnosis and make treatments. the first question comes whether the diagnosis is more important or the treatment is more important. Decide yourself. So assess of delay and correct diagnosis and then correct management for the treatment is very much required for the cancer patient if it is confirmed. So these are the symptoms of the onsets and the discovery of the biomarkers which help for the early detection of the cancer by screening of both the patients in general population as well as the after diagnosis. So this is the approaches for the advancements of primary cancer diagnosis. The early detection is a very important aspect. Then prognosis is dependent, and then response to the therapy. Whether you have to take the non-invasive saliva, then gastric juices or the urines, that will also help, and these are on early trials. So these the genetic signaturings for the circulating tumor cells, that how to approach it for developing some kits or some devices. So first we have to know what is the circulating tumor cells and how it is moving around your blood streams or the lymphatic systems. So this is, and why the circulating tumor cells are become so important for the early detections. These circulating tumor cells, when activated from the primary sites, they enter into the lymphatic systems and they reach to the neighboring organs. Our objective is to early detection of the breast cancer as a model, because the breast cancer is the second largest disease of women's associated with morbidity and mortality. So the early detection of the breast cancer as a model for using a circulating cells, using the blood, providing a promising tool for early detections. The circulating tumor cells can be detected, and characterizations and isolation is a very difficult task by using EMT markers, epithelial mesenchymal transitions markers like SOX, EpiCam and SOX9 to confirm the metastasis of an individual who is suffering from cancer patients. Then we have also characterized through the de novo mutations by Sanger sequencings. So if you will go through the early literatures, you will find that 10 to 20 ml of blood sample has been used for the isolation and characterization of circulating tumor cells. In my mind, it is against the ethics if we will collect the sample cells in such a large quantity from the cancer patients. So we have taken an in vitro biotechnology approaches, and we have given a minimal 0.5 ml of the blood sample is collected and sent for the tissue culture laboratory for more than 72 hours. And we have isolated the circulating tumor cells and compare with the non-tumor cells, although the frequency of the cells are very low. And so using a Ficol gradients method in my laboratory, and we got a very promising results. So you will see this one, the 3D analysis of the stem cells markers, and in this side, right hand you will see the EMT marker expressions. This is a very important slide. This one, lane 2 and 3, the CK19 expression is complete, disappears. So it means the cytokeratin play a very important role during the metastasis. And then we have taken an approach systematically from RT-PCR for MTHFR gene polymorphisms, because that's play a very important role for risk factors. And this is in liver cells from the CTC. This is, again, we got a very important finding that this one of the CK EMT marker is missing. in liver cells. Then we have, there is a concept that the primary tumor cells has been transformed. How it is being activated by the KRAS mutations? Yes. Without activations, they cannot be circulated and they are still remain exist. And it is also been known as a tumor seeds. So when the environment is favorable, it is started or circulating into the whole body streams and affecting the neighboring tissues. So activated by the KRAS mutation. But in my case, the hepatocellular carcinoma, there is no mutation by observed. So systematically, we have also seen the cytogenetic strategies, and we have seen in ALL cases, different kind of the tumor samples, blood streams we have collected, and we have found that what markers will be possible. We have also confirmed in acute lymphoblastic anemia, when we have developed the karyotypes, because the trisomy of the 21 has been associated and confirms increasing the risk of the such type of cancer patient and confirmed by the this fish techniques. Then again, we have seen the EMT markers expressions in the acute lymphoblastic leukemia. You will see this one, there the expression is very high of 573 case 1. So these were further we have characterized by C677 gene polymorphism simultaneously of the MTHFR, which increase the risk factors. This is the miRNA cancer expressions we have found to try to signature over there by using 250 subspecies of the miRNA, but we have seen there is a 3 subspecies are very important for the signatures in the breast cancer patients. So these are the data which showing the statistical analysis with respect to control, if you'll see in 3 coordinates. And because there is a series of the tumor samples of my interest, we shift to the pediatric tumor, that is the Williams tumor. This is one of the most important tumors, and the genesis is still hidden, whether there is a fetal play an important role or the individuals. Because when the baby is born, they is trying to crying, and they says there is some symptoms over the lateral side of the pain. He is putting their hands on the lateral sides because he cannot speak. So when he started of weeping, he is keeping the pain here. So then there is doubt in the parents. Then they goes and contact the clinician, and they found there is a swelling in the lateral side, that is the kidney sides, and confirmed by the tumors. So there is an interaction between the maternal or the fetal itself. So the exact etiology of the Williams tumor is still hidden. But no doubt, there is a genetic and epigenetic factors. So this is one of my interest in this case, and fortunately, we have collected some funding from the DST, and we started our work up to the depth of it, and we have found there's a very interesting nucleotide changes, de novo mutations we have observed beside WT1 and WT2 genes. So no doubt, the genes were activated by the translocation of the chromosomes. So the chromosomal strategy you cannot be forgetted. I will like to request 2 minutes more.
Oh, sure.
Because here I, especially for sharing my work to the young students. So these are the conventional techniques, chromosome, and this, if you see the lower karyotypes, there is a loss of Y chromosomes. So it means there is some sex chromosomes has also been associated with the tumorigenesis. And this is a whole chromosome paintings, and you will find a lot of changes, structural and numerical, you will see it, even though there is an extra copy of the, as a marker chromosome in the right lower one. And one fragile site where we have observed in the X chromosomes. This is a beautiful picture of the WT1 gene mutation in a series of the markers where you will see the arrow there is a loss of mutations. This is WT2 genes. This is specific for the Williams tumor cases. And DNA copy number variations is an increase the risk factor of the disease and genetic susceptibility. So these are my microsatellites marker to study of the loss of heterozygosity. You will find a beautiful picture by showing the extra copy of the bands and by RT-PCR and then copy number variations. And this is the nucleotide sequencing. You will see in boxes there is a change of both frame shift and non-frame shift mutation as well as the single nucleotide polymorphisms. This is WT1, this is WT2. And now we come to the TGF-beta in the Williams tumors. They also shows the either autocrine and paracrine fashion. And we have seen the lane 1 and 2, there is a complete missing of the TGF-beta R1 gene mutation in WT2 cases. And this is the RT-PCR data. And now you will see the DNA sequencing of the TGF-beta R1 genes. And we have found it, the most interesting data is the appearance of stop codons or pre-termination codons. And when we have done the translational research, we have found there is a change of the structure from normal to mutated structure. It means the same drugs when we have given to the patients, it got susceptible towards the either the multi-drug resistance phenomena or due to the mutations where the molecules is failed to bind to the ligand sites. So this is again the MTHFR gene polymorphism, the risk factors either in a homozygous condition or in heterozygous conditions. So this is in breast cancer, then gallbladders, then pancreatic cancer, and then ovarian cancer. I try to cover more of them than the Williams tumors. And the summary. The loss or gain of the chromosome is an important site for chromosomal rearrangements, including when you come from the cytogenetics to the PCR molecular base, then WT1 and WT2 gene mutations, and novel finding is the identification of the stop codons. It means they stop the transcription factors. And the conclusion is the CTC and miRNA offers a promising target for the early detection and prognosis and the therapeutic for the cancer, a scope for developing a new strategy based on gene proteins drug interactions for pharmacogenomics and personalized medicines. The management of the disease of the Williams tumors by supplement of amino acid in the diet as an additional nutritional factor. And this findings was published in Journal of Cancer Science Therapy 7.2 impact factor. I would like to acknowledge to my groups, Dr. Vijayendra, Dr. Pitanjali, Meenakshi, Dr. Veena, and Lovely. So this is my lab was inaugurated by Padam Shri Dr. Laljit Singh of the genetics labs, and these are the hindrance factors. They fail to do your work in lab.